Thank you all for joining today. I'm very happy to have a chance to speak to you all via this nice web platform. I have my disclosures here up on the screen. And just for a bit of introduction, my name is Sam Minot. I'm a microbiome scientist. I work at the Fred Hutch Cancer Research Center. And hopefully you all are scientists or people who are interested in the microbiome, and specifically in the way that we use data to understand what is in the microbiome. And the topic that we're going to be discussing today will be the tools that I've been using to identify microbes, specifically at the strain level, which are associated with human disease. I'm talking about bioinformatics and the analysis of metagenomic data. So this is the topic of metagenomics. This is a type of genome sequencing, which is used for microbiome research. And it's something that has been a part of my work for quite a while now. So starting with my graduate training, I trained under Dr. Rick Bushman at the University of Pennsylvania. And in that work, I used metagenomic sequencing to study the collection of viruses which are found in the human gut. In some of my work after graduating, I spent some time in industry. I worked at a really wonderful San Francisco-based company called OneCodex, which has a cloud-based platform, and the analysis of metagenomic data for microbiome research was a big part of my work there. However, what I'll be talking to you about today has happened since my transition back into academia, where I've come to work at the Fred Hutch Cancer Research Center. Now, this is a research institute. It's located in Seattle, Washington, so in the Pacific Northwest region of the United States. And I work as a staff scientist, and I work in our microbiome research initiative. And so I have this wonderful opportunity to collaborate with a collection of different microbiome researchers, people who are interested in studying different types of biological systems with the common focal point of using microbiome data as part of their research. And so the work that I have is supporting that data analysis. And that's really what's been driving this focus on novel tools for metagenomic analysis. So this is a term that I've been using a couple of times now. So let's take a minute and discuss what is metagenomics and why is it something that we use for microbiome research. So there's a lot that's been written on the idea of metagenomics. But if you want to read in more depth, I recommend this nice review written by Chris Quince and colleagues in 2017. And the idea of this experimental approach is that we want to understand something about what is in the microbiome. What is in this collection of organisms, which is, for example, in the gut, on the skin, in the mouth, these bacteria, viruses, archaea. And one way to profile what is in these communities is to take a physical sample, which consists of a large collection of different cells and organisms, and to extract all of the DNA from all of these organisms. And you may have heard of 16S sequencing, where you focus on a particular gene. Instead, the idea of metagenomics or whole genome shotgun sequencing is that you randomly fragment all of this DNA, and then you take individual pieces and process them for sequencing, high throughput sequencing, generating millions or tens of millions of short sequence fragments, each of which may consist of a couple of hundred of different of nucleotide sequences. And when you do this, each individual sequence fragment could come from any of these organisms, and it could come from any of the portions of their genome. So it's an incredibly rich data type. It's also incredibly large. For an individual sample, you have a FASTQ file, which might be millions or tens of millions of short sequence reads, these short words. For an entire data set, for an experiment, you might have dozens, hundreds, or even thousands of these. And so you really collect an extremely large amount of data about what's in the microbiome. And the challenge becomes, how do you understand it? How do you process it? Now, as I mentioned before, I've been using this type of data in a number of different settings. So in my graduate work, I used this data set to understand what the viruses were that were found in the gut microbiome of healthy humans. These are mostly novel bacteriophage with really exciting biological properties. I've also spent some time in the world of pathogen detection. So using this data type to detect specific bacterial or viral pathogens, which may cause disease. But for today, I'm going to talk about analyzing this data set in the context of microbiome discovery. So here we're thinking about the complete community of microbes that lives on and inside the human body. And we want to identify microbes that might be associated with human health and disease from this extremely large community. So why do we focus on the microbiome? What do we bring to this as a motivation as scientists? And here I'm going to speak for myself. I'm also going to speak for the people that I work closely with, my scientific collaborators, and also the larger worldwide community of microbiome researchers. And here, I just want to make sure that I'm focusing the discussion today on the biomedical research context. There are a lot of different reasons why you might want to study the microbiome, but I really want to make sure that it's clear that here we're talking about the biomedical context. So the idea that we're focused on individuals, on humans who experience a particular health or disease state. They're experiencing some disease, and we really want to, ideally, identify any microbes which might be biologically associated with that health or disease state. We can obtain samples, we can obtain specimens, for example, stool or saliva or skin samples from these individuals. Perhaps we have an experimental design comparing individuals with and without a disease state, or something a bit more complex experimental design. But in this process, I am a computational biologist. I spend my time on the computer, and so I'm focused on the way in which we analyze the data that is obtained from these microbiome specimens. And the goal in this context for this data analysis is that we'd like to propose a biological mechanism for the role of the microbiome in human health and disease. But I think it's not enough to merely propose a hypothesis. We also have to make sure that that hypothesis can be tested, that these are testable hypotheses. And with this goal in mind, the research, the work that I do is really focused on this analysis of metagenomic data in a way in which at the end we result in a specific reagent. So it could be a culturable microbial isolate. It could be a purified protein or a small molecule. We have some proposal of a specific reagent which can be used to test the proposed the biological mechanism, that these are falsifiable hypotheses, that these physical reagents can be used in a controlled experimental setting, for example, a preclinical model. And the reason that we focus on these falsifiable hypotheses is that we believe this is how we can move forward towards developing some form of microbiome-based therapeutic, where the knowledge that we gain can be ultimately used to treat, to prevent human health and to improve human health and disease. So this is the reason why we study the microbiome. Now, how do people actually process the data that results from metagenomic sequencing? What are the approaches that people take? Since I've been working in this area for a little while, I tend to think about breaking these analytical approaches apart based on the ontology. So based on the system of knowledge that you use to organize information. And I think the one that might be closest to mind for everyone might be taxonomic analysis. So if you've worked with metagenomic data, you might think about Kraken or Metaflan2. These are very popular, and Metaflan in particular was a transformational tool in my own work, maybe about a decade ago. And the idea here is that we're able to analyze metagenomic data to quantify the abundance of organisms based on the names that we've annotated on the tree of life. The idea of a taxonomic tree is that all life can be placed on a bifurcating tree. We can give names to each of these different leaves and nodes, and that is what we are quantifying when we are analyzing metagenomic data in this mode. Now, I think that this is extremely powerful, and I think that it works in a lot of cases. But as a microbiologist, I'm also aware that there are fundamental biological limitations to the notion of taxonomy. And this is because there is non-taxonomic diversity of bacteria. So, for example, if we were to summarize the diversity, the genomic diversity of a collection of isolates of Bacteroides theta iota omicron, or B theta, we can summarize them based on the collection of genes which are found in their genome, grouped together based on the genes that are found together. And we find that, there are a collection of genes which are found across all B theta. This is, for example, the core genome of B theta. But when we look at the other elements that may be found in the genome, different plasmids, genetic islands, or operons, we can see that these particular genetic elements are found in the organism, in the specific isolates shown with these blue boxes here. They're found in groups of isolates which cannot be distinguished or cannot be summarized based on any particular phylogenetic or taxonomic tree. You cannot draw a tree which captures the contribution of each of these three different boxed genetic elements independently. And the reason for this is biologically extremely understandable. Bacteria are not the asexual, obligately clonal organisms that the notion of taxonomy assumes them to be. This is because some genetic elements are inherently mobile, such as plasmids and phages. Other genetic elements can be exchanged through forms of sexual reproduction, such as natural competence and other biological mechanisms, which means that there is genetic diversity in bacteria which cannot conform to any possible taxonomy or any single phylogeny. This is not an observation that's unique to me. This is something that, in fact, you see anytime you compare the pangenome of a collection of bacterial isolates. There's a portion of the genome we think of as the core genome per species, and then there are accessory genetic elements, and the distribution of accessory genetic elements is not inherently clonal. So with that caveat in mind, I'll say that taxonomic analysis is very useful. However, it does not encompass everything that we need to know about what bacteria can be in the microbiome. Another approach that is extremely popular and I think has a lot of value is the idea of analyzing metabolic or really functional content. So instead of the names of organisms, we might be interested in whether or not they can metabolize a certain biological transformation or step in the metabolic pathway. And I think Human 2 from the Huttenhauer group. So, as I mentioned earlier, Kraken, developed by Derek Wood and colleagues, Metafon 2, Nicholas Agata and the Huttenhauer group, as well as Human 2. And I think this is an area that's extremely valuable. It's also limited by the limited annotations that we have available. Another approach that I think of as this genome resolved metagenomics was really pioneered by Jillian Banfield's group at University of California, Berkeley, as well as, I'd say, the really fantastic Envio tool developed by Murat Aran and his group at the University of Chicago. And in this approach, you might think about summarizing what's in the microbiome based on these reconstructed complete genomes, which you're able to derive by de novo assembly from individual metagenomic samples. Now, there's another approach that I want to focus on today, and this is gene-level metagenomics. So using the gene as the fundamental unit of analysis. And there is a tool that's been around for a while, MGRAST, which has this general idea, but instead I'm actually going to focus on the area that I've been spending time on, which is this tool that we've called Geneshot, so gene-level analysis of shotgun metagenomic sequencing data. And the goal here is really to use genes as a way to unify all of these different ontologies, as a way of reaching across these different ways of organizing knowledge. And the reason that we have for approaching genes in this way is that if you look at one particular microbial gene, you can assign it a taxonomic annotation, you can assign it a metabolic or functional annotation, what does this gene do, and you can also align it against a particular genome. Is it present in one genome or another? And for that reason, I think that genes can be extremely useful ways of approaching the ways that we as scientists try to get some understanding of what is in a particular, what is an association with human health and disease might be. Now, since I was going about the process of trying to develop some novel tooling, this approach, I had the advantage of having this wonderful group of collaborating scientists here at Fred Hutch, where I was able to work with them on their scientific projects and get some general overview idea about what would be useful to provide to these researchers and also to microbiome researchers in general. And this is going to be more of a sort of a gestalt here, but the idea is that if you performing this microbiome analysis, you might be interested in identifying microbial taxa, so taxa being sort of taxonomic entities, species, genera, phyla, etc. Identifying microbial taxa, predicting the biological functions that are associated in the microbiome. I also felt like there could be an opportunity to provide automated statistical analysis. So not asking the researcher to do everything in the world of statistical analysis, but providing a foundation based on the experimental design that they provide, as well as integrating with the comparison to reference genomes. Because oftentimes you can end up performing a microbiome experiment and then wanting to go back to the freezer to identify an isolate, which can be used to test a particular prediction. Finally, I found that this type of data ended up being extremely complex. There's many different things that you can, different ways, different dimensions that you can summarize, and that this interactive visualization was something that I thought could be useful to make this entire approach a bit more tractable. So when you look across the types of tools that I've mentioned before, I'd say that they all do some level of these individual things quite well. You can really definitely learn a lot from these successes. And so the goal here was to develop a tool or a collection of tools which was able to encompass all of these within sort of a unified framework. And so in this project, and this is what I'll be discussing today, is Geneshot. So this is a gene-level metagenomics or shotgun sequencing pipeline, so analysis pipeline. And this is a tool that I've developed in very close collaboration with Dr. Jonathan Golub, a physician scientist at the University of Michigan, as well as Dr. Amy Willis, a professor of biostatistics at the University of Washington. Rather than walking you through every step in the pipeline, which I think would be only interesting for a very small number of people in the audience, instead I'll just show you this nice schematic diagram put together by Carolyn Caspin in order to walk you through the idea of what this pipeline does. Once I finish with that, I'm going to show you some results from an actual data set, walk you through what we can learn using this approach. And finally, I'm going to point you to the resources that you can use to run this type of analysis yourself on your own data. And so that's how we're going to spend the remainder of our time here today. So in processing microbiome metagenomic data through this Geneshot gene-level analysis pipeline, the first thing that happens is that we build a de novo gene catalog. We want to identify the genes, the microbial genes that are present. So reads are trimmed and filtered for host DNA, and then each individual specimen is processed for de novo assembly. We reconstruct contigs from the raw sequences. The protein coding sequences from each contig are then identified and then deduplicated using LinClust across all the different specimens. This gives us a non-redundant gene catalog from a particular experiment, which forms the unit of analysis for all further downstream applications. Each of these individual genes, as I mentioned before, can be annotated for predicted metabolic or biological function. In this case, we're using the eggnog algorithm. We can also assign a taxonomy for each individual gene. In this case, we're doing it by alignment against the RefSeq genome collection using Diamond. So now we have our gene catalog. When you do a typical microbiome experiment, this could easily be millions of unique genes, looking at even clustering at 90% amino acid identity. And I think this is really the point at which gene-level analysis had stayed as an intractable approach previously. If I have millions of genes and I have dozens or hundreds of samples, the dimensionality challenge is really too large for making any sort of robust statistical inference. To mitigate this challenge of dimensionality, the next step in the process is to group genes by co-abundance. After constructing this gene catalog, we're able to quantify the abundance of every gene in every sample. The notion of abundance here is really the relative abundance of organisms which contain this gene in their genome. That's what I mean when I say gene level abundance. And we wanted to group these genes by co-abundance based on a proposal, an idea from this paper. So this was Nielsen et al. In 2014. And what these authors really described was a really wonderful concept, which is that if you end up looking through a set of microbiome samples, so in the top left, right, we have specimens from a number of different individuals. If you observe that a set of genes are consistently found at similar abundance across similar samples, the biological interpretation is that those genes were encoded on the same piece of DNA or on the piece of DNA that are co-abundant from either co-abundant species or found in the same cell or strain across these individuals. The idea here is that if you then cluster genes by co-abundance, you will form these co-abundant gene groups, or CAGs. CAGs is the vocabulary word for this particular presentation. There's no test at the end, but this is the vocabulary word I want to leave you with. A CAG is a co-abundant gene group, and the biological interpretation of a CAG, based on this wonderful paper from Nielsen et al., is that a CAG can be the core genome of a species, always found in the same cell. It also might be any sort of accessory genetic element, a mobily transferred plasmid, a genetic island, an operon, a phage, for example. All of these will form their own independent CAGs because they are found at consistently found at similar relative abundances. Now, the process of identifying CAGs is, in an exhaustive sense, incredibly difficult. If you have millions of genes and you want to do clustering based on co-abundance, that would require you to perform trillions of pairwise comparisons, which is not practical. In tackling this problem first, I published a paper with Dr. Willis last year where we described how you can use this ANN algorithm, or the approximate nearest neighbor algorithm, in order to speed up this process and make it more computationally tractable to go through and identify CAGs from microbiome experiments. And that's some code that's really one of the key pieces of Geneshot. So now that we've gone through and we've grouped genes by co-abundance, now we have a much smaller number of features, where each feature, each CAG, is the core genome of a species, a set of co-abundant organisms, or an accessory genetic element. And then we can use those features to do things like calculate dataset associations, perform statistics. As part of Geneshot, when you provide the starting place, you provide it with the raw FASTQ data, but you can also provide it with the experimental design. For example, treatment versus control, time point, etc. And you can provide a formula which is then used to test for the association of the abundance of each CAG with that experimental design using this corn-cob algorithm developed by Dr. Willis and colleagues. Now, as a result of all of this pipeline, all of this data processing, there's really an immense amount of information that's provided. And really all of the data is then summarized and provided back to the user who ran everything, which provides a wonderful foundation for biological insight, which I'm going to walk you through right now. Now, I've described this as a pipeline. It's also useful to note for the bioinformaticians in the group what we mean when we say a pipeline here. So you can go to this URL, and I'll put this up as my final slide, which has all the code needed to run Geneshot. And this is code which is run using the Nextflow workflow management system. And I think that the reason for using a workflow management system is that when you actually run compute as a bioinformatician, you may run it on a number of different architectures. So you might be running analysis on your laptop. You might have a tower that's underneath your desk that's running everything. More typically, you run it on a high-performance computing system at your institution. For example, Slurm, PBS, some sort of HPC. You might also be interested in using cloud computing. So for the work that I do, I tend to run everything that I do on AWS, which is extremely convenient for me. And using Nextflow, you're able to configure all of that independently from the workflow. So you can set up your own system with Nextflow, just set up your own, tell it what type of configuration you're using, and that allows you to run any workflow written in this language on your specific compute infrastructure. And that means that in order for you to run Geneshot at your own in your own lab or your own place, you can just configure Nextflow once, and then that allows you to run Geneshot for yourself. So now that I've told you what Geneshot is, the idea behind it, how it generally works. Next, I want to walk you through some example data. So I thought that for the purposes of these types of talks, it would be helpful to present some data from a real microbiome experiment. In this case, I'm showing you data from this study, that Nature Medicine paper from March, in which they tested the effect of a controlled feeding experiment, so underfeeding, on healthy humans. They also had, you can see in the title, they mentioned oral vancomycin. I'm not going to describe that arm of the study, but here we're really focused on underfeeding. And they did so, one reason I use this is that it had really fantastic experimental design where they were able to administer a controlled diet in two different arms. Healthy humans participated in this study. They were housed in a controlled setting. They were fed a controlled diet, so they're 100% of their recommended caloric intake for 4 days to establish a baseline. A sample was taken at the end of those 4 days. And then for the next, so days 5, 6, 7, one study arm was underfed, so the caloric intake was reduced by 50% for those 3 days, and another specimen was taken at the end of those 3 days. Then there was a washout period for the next 3 days, and then they were overfed. So we had this crossover design. So we had decrease of 50%, then an increase by 50%, with metagenomic data being taken at the end of each of these 4 periods, not counting the final period here. In addition, there was a second study arm in which the same approach was taken with the opposite order of administration. So first overfeeding and then underfeeding. The goal of the scientists was to identify which microbes consistently changed in relative abundance as a result of this controlled intervention of reduced or increased caloric intake. And they were able to identify a handful of species, so Akkermansia, Allostypes, Barnesiella, which they identified as significantly increased, mostly increased in the underfeeding arm, as well as one that was increased in the overfeeding arm. And so the goal for my analysis was to analyze their published data, which they published with a really nicely described experimental design, as well as a good metadata sheet. And that allowed us to reanalyze the data in order to address the exact same goals that they had of identifying organisms that were associated with this caloric intake, overfeeding and underfeeding. So let's walk through the results. What did we get from Geneshot? So the first thing we did was build a gene catalog. So we were able to perform de novo assembly and assemble a couple of 100,000 of genes from each individual sample. After deduplicating and then quantifying the abundance across all samples, we found that of the, I think it was 2 million genes across the entire gene catalog, when you don't require complete coverage based on assembly and instead use a much more sensitive form of alignment-based detection, we're able to detect much more. Up to 1 million genes for each biological specimen, with some relationship to sequencing depth. Now, the point of this that I want to emphasize is that for every individual metagenomic sequencing read, so here we're talking about tens of millions of reads, we were able to uniquely assign by alignment around 80% of the data of each individual sequence fragment to a single protein coding gene from this gene catalog. Now, when you think that only 90% of a bacterial genome is thought to consist of protein coding sequences, the fact that we're able to capture around 80% of the reads in these uniquely assigned genes, I think is really encouraging, because it means that we're able to really capture a large amount of the raw data of what the organisms are in every specimen. Now, the next step, as I mentioned, was to take these abundances and group genes by co-abundance into CAGs. We can see that there were 2.5 million genes in the catalog, and these were condensed into a little over 250,000 CAGs. We can look at the size distribution of these CAGs and the number of genes found in each bin. And we see that there are hundreds of genes which are of the size you'd expect from the core genome of a species. There's also a number which are likely to be co-abundant species that are found together in the same samples, as well as a large number of CAGs of the size you'd expect from accessory genetic elements, such as operons, plasmids, phages, etc. With this process, we're able to reduce the dimensionality by an order of magnitude. We had 2.5 million genes, and we were able to identify 200,000 CAGs. This means that we have many fewer features that we need to test for association with the experimental design. And so the amount of dimensionality challenge was mitigated quite considerably. So the next step was to identify associations with feeding. This was actually set up before any of the compute took place. When the run was started, we did so with a metadata sheet where every individual specimen in a table, an Excel spreadsheet, and a CSV had a column for host diet, which I'm showing here. And the values there were either for the control diet, for the decrease, or the increase diet based on what section of the experimental design it was in. Then when Geneshot was run, we could provide a single flag where we indicated which column. This could be a univariate. It also could be a multivariate formula. In this case, it was univariate design, where we provided this categorical label as the substrate for the statistical analysis. As a result of providing this one flag, Geneshot used the corncob algorithm to compare the relative abundance of every individual microbe that was detected as a function, comparing between the control and underfeeding, and then independently comparing the control and the overfeeding. As a result, in the output of Geneshot, we have tables which provide us with these estimates of associations, which I'm plotting here as a volcano plot. So on the left, we have the association with overfeeding. On the right, we have the association with underfeeding. On the vertical axis, we have the FDR corrected p-value or q-value, so the negative log 10. And then on the horizontal axis, we have the estimated coefficient. So points further to the left were CAGs that are more abundant in the control diet. And on the right, we have CAGs which are more abundant in either the underfeeding or the overfeeding. And so this automated statistical analysis is able to tell us which of the microbes, which of the CAGs are most consistently associated with this experimental design. Again, this is without having to do any custom code. So let's look at a little bit of what these results are. So every point is a CAG. Let's look at one of these. So here's a CAG, which is near the top, so it's rather significantly associated, it's rather consistently associated. And when we look at the relative abundance values which resulted in this particular Q value, we can look at the abundance of this CAG across different specimens. Each colored line is a participant. Each panel is a study arm. So on the left, we have the underfeeding first. On the right, we have study arm 2 with overfeeding first. And this association indicates that the relative abundance of this CAG is higher in the underfeeding samples compared to the controlled diet. Now, this is not where the analysis ends because we have a lot more information about what is in this CAG. What is this CAG in particular? For example, we can look at the taxonomic annotation. So this is a sunburst plot where the amount of the fraction of the circle, which has a particular organism's name, is the fraction of genes which were given that annotation. We can see that a large number of these genes were just annotated as Clostridialis with nothing more specific, but there was the largest fraction of the genes were annotated as Enterobacterium hollii, which is an organism which is known to be found in the human gut and has been identified in a number of different studies. Now, instead of looking at a single CAG, the way that we get a better understanding is by summarizing the composition of all the CAGs which are at this top portion of the volcano plot. So let's look at the collection of CAGs which are most consistently associated with underfeeding. And we can do so with this taxonomic annotation. So here we are summarizing the taxonomic abundance, the taxonomic annotation of a collection of CAGs, which are all most consistently associated, enriched in underfeeding. In this heat map on the left, every column is a CAG, and every row is a taxonomic label. The color in each box indicates the percent of the gene annotations which are consistent with that particular taxonomic grouping. So blue means more annotations. With this type of display, we can pick out this Enterobacterium hollii, and we can see that not only is there a single CAG, which has this blue box of annotation for Enterobacterium hollii, in fact, we can see that there is a collection of CAGs. There's more than one CAG, which all have this same annotation for the same species. And this starts to build more evidence for us that there is a true biological association of this organism in increase with underfeeding. when we group together these tags based on their annotation as A. hollii, we can summarize their abundance across the exact same experiment. And here you can see that in all but 2 of the participants, we can see a dramatic increase in the relative abundance of the organisms containing these 5 tags, which are annotated as A. hollii, in the underfeeding. Now, this starts to build even more evidence that there's a true biological role in the increase of A. hollii as a result of underfeeding. Now, taxonomic annotation is not the only thing not the only type of data that we have access to with this type of approach. So I mentioned earlier that we had this biological functions. So in the same way that we can inspect the taxonomic annotation of these underfeeding associated tags, we can also inspect the functions that are predicted to be encoded by these genes. And this is the great thing about using a gene as your primary unit of analysis, is that we have this direct correspondence of the fundamental unit between taxonomy and function. Here we can pick out individual functions, for example, which are related to nutrient sensing and transport, which helps inform a mechanistic hypothesis of why a particular organism might grow to higher abundance as nutrient availability decreases. Now, finally, I mentioned that there was an interest in comparison to reference genomes, specifically thinking about identifying culturable isolates for validation experiments. In order to demonstrate a little bit of this functionality, we took all of the genes from this gene catalog, and we were able to align them against a collection of reference genomes. So you can provide any sort of collection of reference genomes. Here we use a subset of the NCBI reference genome database. And in the same way that we looked at earlier, we can summarize the similarity of these underfeeding associated tags to a collection of reference genomes. So here, every row in the heat map is, in fact, an assembly. It is an isolate which has been assembled and has been deposited into RefSeq. Now, there's a lot of detail here, and I don't have time to walk through all of it, but I do want to mention a couple of key features here. So the blue boxes indicate when a particular tag, a particular set of genes have been aligned to. They have significant similarity by amino acid alignment, translated alignment using diamond against a particular assembly. So we can see a couple of different patterns here. So here we have a tag, which is found across 3 different isolates across Prevotella. This unit of analysis, the tag, appears to be shared across the genus Prevotella from this analysis as well. However, we're able to distinguish it from all the other elements that are shared across Prevotella because we're using the gene as the unit of analysis and not the taxonomy, which I think is really wonderful. In addition, you can see that there are other groups of genes which are found disparately, so here across 4 different isolates, and these 4 isolates, the only thing in common between them taxonomically is actually the phyla. And this is believable given that some genetic elements are actually able to move, like broad-range plasmids, are able to move and be found across incredibly distinct or taxonomically distributed organisms. We also are able to identify, here in the box I'm showing one of these references on the left. With this approach, we're able to identify organisms which don't have a particular taxonomic assignment. So this is an allocytes with a species name, which is a particular isolate which is not given a recognized species name, and this biological signature of this uncharacterized organism is still included in the analysis because when we do our gene level metagenomics, we're not requiring that there is that we're not conforming to the taxonomic tree in order to pull out these biological signals. And finally, when you see this sort of complex overlapping pattern of the identity of gene groups within different isolates, you can see that there is this complex relationship which would have been completely lost if we'd been using the taxonomy as the fundamental, like, ontological basis of our approach. So the final thing I'll say about this particular slide is, with this approach, we're also able to identify specific isolates which contain a large number of these underfeeding associated genes. Now, this is actually a reference. This is from RefSeq. This is not an organism that I have in the freezer. But if you did run this against your own isolate collection, instead of running it against RefSeq, you would then be able to go back directly to the tube in the freezer that contains this organism and say, here is a physical reagent, which I could use, I could grow it, I could use it in a preclinical model, and I could try to reproduce the findings from this human-based microbiome experiment. I now have a reagent which I can use to test the model that I am proposing, and also in this system, then refine down, perhaps by inspecting where these genes are in the genome, to identify the biological process which might underlie this. So I'm clearly very excited about this. This is something that I'm finding extremely useful for my work. From the practical standpoint, I want to really make sure that I make it clear that there's actually a very small collection of tools that we've broken apart here, which in my attempt to make this more tractable and useful for a broad audience. So on the one hand, we have Geneshot. This is the tool where you provide FASTQ files, your experimental metadata sheet, a formula if you have it for an experimental design, and it'll go through the entire process of de novo assembly, quantifying the abundance of genes, forming tags, performing, annotating all the different functions and taxonomies, and producing this copious amounts of data, which then are available for inspection. The process of comparing the results of Geneshot against a catalog of microbial genomes is actually provided with a separate code base. So this is the analysis of microbial genomes for microbiome analysis, or AMGMA, which is a palindrome, which I enjoy. And this is actually a separate code base. I'm going to show you a link to all three of these tools in just a second. And this was separated out because you might want to go about aligning against different sets of genomes many different times. And if you added one more genome to your isolate database, I wouldn't want you to have to start from the raw FASTQs. And so that's why these things have been pulled apart a bit. Now, finally, you can see there's an empty space here. When I was personally going through and inspecting all these Geneshot results, I kept finding that I was spending a lot of time in Jupyter, in a Jupyter notebook, writing Python code to make these plots. And I was sort of making the same plots again and again, and it was taking me quite a while to go through and provide all these different types of outputs for my collaborators. And I finally broke down and decided to try to make an interactive visualization tool. And so here we have, this is something that is, I'd say it's a working tool, this GLAM browser. So this is the gene level analysis of metagenomes browser. And the GLAM browser will provide visualizations for all of these results. It's really a massive amount of data. We're talking about millions of genes with statistical associations across multiple different experimental designs and maybe hundreds of thousands of microbial isolates. To pull this all together and make it accessible, this is why I've put together this GLAM browser, which is also available for use. Now, what I'm showing you here next is a short video. I decided that it would not be feasible to try to do a live demo in this platform, but I'm going to try to play this video right now. And here you see an example of me interacting with this data set. And the sort of, the big reveal here is that most of the plots that I showed you, I actually generated using this platform. This is the most useful way for me to get a quick idea about what happened in a microbiome experiment. And so with these types of annotation plots, oftentimes, it's not enough to just generate a single one. You might want to inspect individual parts of it. You might want to filter. You might want to show fewer or more different tags and inspect the results in different ways. And that's what this interactive browser provides, where you can customize these displays and then download the figures in order to present them in the subject for publication or for forums like these. So this is a set of tools which I'm finding extremely useful for my own work, which I hope that you might also find useful for yours. I also think that they might provide a general idea, a framework for how you can approach metagenomic analysis in a way that's really, it's really helpful for microbiome research. And so if other people end up getting interested in this gene level analysis, I think there's a lot of opportunity to keep developing these types of tools for analysis, different ways of visualizing the data that are maybe building off of this in more and better ways. So I'm going to, I want to make sure to acknowledge my collaborators, the sources of funding, including from the NIH, the U.S. National Institutes of Health, as well as my institution. We also had some support from the Amazon Web Service and their Spot team for some of the work doing this development. And I'm going to leave this slide up with a link to these computational tools, which are publicly, you can access these URLs right now. There's actually there's wikis on all of these where you can get a few more details about how to go about doing this type of work yourself. And so with that, I'm actually going to stop the presentation. I'll say thank you to everyone. And now I will be happy to take questions.
Thanks for your presentation, Sam. As you just said, it is now time for the Q&A. To ask Sam a question, all you need to do is type it in where it says ask a question and then press submit. So Sam, our first question asks, how complete and accurate do you feel the genomic reference databases are for the gut microbes?
This is a question that I struggle with on a daily basis. When I compare the genes that you derive by de novo assembly from a particular microbiome experiment with the genomic reference database that are available, and here we'll just talk about RefSeq, I find that there's really quite a large range. When you identify organisms, without any culture, so culture-free methods, you'll find that there's a number of organisms that are found, for example, in the human gut, which are recognizable. For example, we can tell that they might be Clostridia or Clostridialis, but when you try to find the best match for the actual isolate or the actual species, oftentimes we find that there's really nothing that's all that similar. In some other presentations that I've given, I've shown a bit more systematic summary of this. But I think that when we get down to 80% amino acid identity as an arbitrary cutoff, we're really looking at a minority of the organisms that are truly found in the human gut, which have a match in reference databases. Now, this is always changing. We're getting better and better and better at making these reference genome databases, and we're also much better at identifying some taxa than others. And so it's definitely a moving target, but I think there's a huge opportunity in being able to sequence more isolates from your area of interest and using that to leverage even more information. I think there's a lot to gain by continuing to expand our reference genome collections.
Excellent. Thanks, Sam. Our next question asks, what are the best ways to limit the number of host sequences that often swamp out pathogen DNA in shotgun metagenomics?
The approach that we've taken for host removal is one that I think is best utilized in not particularly host-rich samples, so such as stool. In stool, when you isolate DNA from a healthy person who is not experiencing, large amounts of bleeding or sloughing or anything like that, we find that the vast majority of sequences are non-human. As part of Geneshot, we allow the user to provide their own host genome. And the first thing that happens is that reads are aligned against this host genome, and anything that aligns to the host genome is removed. So we perform host removal as really the first thing. All data is immediately filtered by that. I think this works relatively well when host DNA is not the majority, as you might expect from some body sites or from some sampling methodologies. And it also works well when you have a good reference genome for the host. The other point to consider is that you can account for this in your experimental design. If you know that a group of samples has more host DNA than another, you can include that as a parameter in your model and specifically identify that contamination as part of the automated analysis in Geneshot. So I think a combination of very close inspection of your sampling site, some knowledge of your host organism, and then also incorporating potential contaminants into the statistical model all can be used to mitigate this problem of host contamination quite well.
Excellent. Thanks, Sam. Our next question asks, how do you handle genes that are expressed by multiple bacteria? Do they count for one bacterium or any bacterium that could produce it?
Capturing genes that are found across multiple bacteria is something that I really find to be the most exciting feature of this approach using Geneshot. In a practical sense, the way this is achieved is that every protein coding sequence from every contig is clustered by amino acid identity. By default, this is done at 90% amino acid identity. If you have if you have a gene, which is found in two different organisms, and they are more than 90% similar to each other at the amino acid level, then they will be grouped together and considered to be the same gene for the purpose of this analysis. This is a parameter which can be customized, and I have some collaborators who have explored this parameter space. I think that exactly what number is correct is something that could be explored even further, and I'd be interested to see more systematic surveys of it. But by combining these genes into a single unit of analysis, I think that's really how this analysis approach is distinguished from the genome-resolved metagenomics approach. If we were just going about and reassembling every genome, then two different strains with the same gene, which were 95% similar on the amino acid level, those could easily be represented into two different experimental units or units of measurement. But by clustering the genes together by amino acid identity, we're then able to say, well, we think that these are probably performing the same function, and so we'd like to treat them as the same thing for our analysis. And so that's how we operationally consider that problem for the Geneshot use case.
Excellent. Thanks, Sam. Our next question asks, what is the minimum or optimum number of reads that you need to have to build the de novo gene catalog?
In order to build a gene catalog which captures the information that's present in a sample, you need to have a combination of deep enough sequencing across individual samples, but also you can get a lot of value by having a breadth of sequencing across many different samples. What I mean here is that in order for a gene to be included in the catalog, it only needs to be present above the limit of detection, which is usually about 3 to 5-fold depth of sequencing in at least one sample. So even if a gene is below the limit of detection for assembly in one particular sample, so for example, you have 10 million reads, but you only have 5 reads from this one gene, you wouldn't be able to detect it by assembly. However, if you were, I'm getting some feedback here, when you perform assembly across many different samples, you might find that it's above the limit of detection in another sample. In a practical sense, for microbe-rich environments, I would recommend millions of reads, maybe 10 million reads per sample. But I think that a lower depth of sequencing can also be really informative, particularly if you have large amounts of specimens in your experiment. So maybe from, instead of a dozen, having more like 100 or something like that.
Excellent. Thanks, Sam. Our next question asks, using CAGs, how does the treatment and control samples, sorry, let me start that one over again. Using CAGs, how does the treatment and control samples, how can they be differentiated?
The approach here really provides the user with any number of opportunities for labeling their samples. When you provide the inputs to Geneshot, you provide a set of FASTQ files, and you also provide a CSV or a table. You can include any number of different columns in that. You might have multiple different types of controls. You might want to categorize a single sample in many different ways. You can do that with as much different types of labels as you find useful. All of those annotations will be carried forward throughout the entire process, and you can include any of them in the statistical model that you provide, which summarizes your experimental design. We did this in order to provide as much flexibility as possible to the user, That always comes at a cost when you provide this flexibility, but we've generally found that as long as you don't do something, particularly egregious, then this is able to provide the user is able to then sort of pick whatever's best for the way that they're approaching their science.
Excellent. Thanks, Sam. Our next question asks, can Geneshot be used for environmental samples?
As I mentioned at the top, my focus is on biomedical research, but I'm acutely aware that this methodology of metagenomics is something that also has a lot of utility for environmental analysis. I think that using this exact same methodological approach could have lots of utility for environmental samples. And I also think that it may provide a lot of benefits, especially in settings where organisms may have been cultured less, when we may have less of an idea of what the taxonomic tree of life is for these particular organisms, or even reference to these access to reference genomes for these organisms, for example, from a soil sample. I would also say that based on my experience, I know that some of the initial steps, such as de novo assembly, can be much more challenging in the context of something like soil or water samples. And so I would be interested in hearing more from users about their experience. I do think that some additional level of sequencing, as well as additional time or compute costs associated with assembly, will likely be required. But this likely will depend on exactly which environmental setting you end up studying, and the complexity, the number of distinct organisms, and the evenness of distribution in that particular setting. But at its core conceptual level, there is nothing inherently biomedical about this software set of resources.